Hello everyone, welcome tonight to the MIT Museum. I'm Jennifer Novotny and I'm the programs coordinator here. I'm really glad you all were willing to join us tonight for our second part of our soapbox series. Just so everyone knows, on your chairs there should have been a flyer for our soapbox. It lists all 4 talks we're having. So you already missed our first one, which was from DNA to designer genomes, but hopefully you'll join us for the next 2 Wednesdays. We have on October 14th and October 21st for the next 2 soapbox series about synthetic biology. The format for tonight's program is a little different than what a lot of people are used to. So we're going to have 2 short talks by Peter Carr and Jeffrey Way. They're going to be about 20 minutes each. And then we're going to have small group discussions at your table. We're going to hand out iPads for people to tweet in your own questions about your discussion, and we'll have a Twitter fall posting all of the tweets that everyone sends in. And then we're going to have a large group discussion with question and answers with questions from the audience, from our online audience, so we have a live stream going, as well as questions through Twitter. So hopefully this will be a great opportunity for us to have a nice discussion and conversation about synthetic biology. And so with that, I'd like to welcome our first speaker, Peter Carr, who is a senior scientist at MIT Lincoln Laboratory.
Thank you. Am I coming through okay? Everybody hear me? Okay. So, being next to MIT and here in Cambridge, I don't want to make any assumptions about our distribution of backgrounds and perspectives in the audience, although I wouldn't be surprised if there's a number of university-affiliated folks here. Let me just get it, if people are willing to humor me by a show of hands. Who in this audience considers themselves currently affiliated with one of the local colleges or universities? I'm glad to see that it's in the vicinity of half, and not everybody and not nobody. I'm glad to see we've got a spread in the room. There's a lot of other relevant questions that I could ask. Probably the one I'll try. Who here has spent, by their own estimation, a significant amount of time studying something along the biological sciences? All right, again, a mix, but we've got a fair amount of background. And that certainly helps me tune the things that I'm going to attempt to say. So when we were given the challenge of thinking about the SynBio barrier, we were given a lot of latitude to what are our ideas about the barriers that the field of synthetic biology experiences or may experience, may be afraid of experiencing as we're looking ahead into the future, as well as lessons learned from the past. And so what I've done is attempt to sample some of my thoughts here without pretending that these are the be-all and end-all of all of the limits. They are some of the ones that are, they're some of the barriers and challenges that are on my mind and that I see from my perspective. And so I like to put something up in a general sense of, and I didn't see if we had a pointer here, I apologize. Anyway, it's not urgent, but these are just a few examples of things that we use living creatures, living systems for. We eat, we build, we burn, we need energy, that is. There we go. So the ones on the top are kind of generally along the lines of making things that we need and use. And one of the hopes is through genetic engineering, we can replace some of the things that we need or want that are or processes for getting them that otherwise are harder on the environment, or more costly, involve harsher chemicals and so on. And then in some of my work, and in some of the aspirations of synthetic biology, we have these other categories of what living systems can do in different ways, including detecting problems, performing chemistry that instead of making things that we want, gets rid of something that we don't want. And there are living things that can help fight off disease in different ways. Sometimes that's by making medicine, and it's kind of illustrated here, but there are other more complex therapies, including ones that through genetic engineering we hope to attain. And so one of the things that comes up frequently and maybe on some people's minds, depending on their background, there's a lot of different overlapping definitions of synthetic biology, and so I'm helping to frame my perspective on that which is through the window of the different ways that humans have manipulated living systems over the millennia, starting not surprisingly with old school, and old school is still very powerful, selective breeding and other kinds of conventional plant and animal breeding that have given us a lot of function. That have given us high crop yields, that have given us dogs that can be trained to sniff out particular things. You go from drugs to cancer to, but we get a lot of great functions through selective breeding. But of course, it's slow. It's very laborious, but it's still powerful. When you look at the sorts of innovations that people started coming up with a few decades ago, in the 1970s are a good example, where there were several advances in how we manipulate the DNA of living things and how we, in particular, can cut and paste, if you will, the genetic material, DNA, from one organism into another, transfer a function from one organism to another. You can kind of think of this as classic genetic engineering. And throughout most of its history, the focus has been on single genes, single functions, and that's very powerful. We don't want to, if we try to draw a line anywhere between genetic engineering and synthetic biology, we clearly don't want to discount the power of simplicity. So shown up here, cartoon-wise, is the idea of green fluorescent protein taken from a kind of jellyfish. It's on jellyfish, but it was in the picture. To light up the biological secrets, if you will, of diverse kinds of living organisms. So the green fluorescent protein, and now numerous variants on it, have been an incredible research tool, and that's why the technology is won the Nobel Prize. The Nobel Prizes are coming out right now, I'm excited about it as well. But this is essentially what you can do, cutting and pasting a single function from one organism to another. And then one of real medical significance, the idea that for insulin for treating diabetics, traditionally was a byproduct of the meat industries, of bovine insulin, isolated, but never in the kinds of amounts that people really needed. But one of the early biotech successes was the cloning of the gene for human insulin into bacteria so that you could produce a valuable, useful human insulin in what are basically microbial factories. So you could grow those up in a large container, and through genetic engineering, they would be producing the thing you wanted. You would crack them open, purify it, and you would have an extensive supply now of insulin. And so the contrast I want to draw with synthetic biology is not that there's some crystal clear, sharp line between genetic engineering and synthetic biology. I think most people would say there's a lot of blurring there. But some things that are worth identifying within my own experience are complexity and ambition. And so diagrammed up here in a visual lingo that I won't be explaining, but essentially each of these visual elements is a genetic element. It represents a piece of function encoded in DNA. And this is a circuit from Chris Voigt's lab. And this is a fairly sophisticated genetic circuit involving dozens of different pieces of DNA, some of which have come from many different organisms and been pieced together to perform what is complex logic by biological standards, simple logic by electronic standards. So we're not trying to necessarily replace electronic computation computers with this type of function. This is in sharp contrast with single functions or small number of functions that we've normally seen in genetic engineering, as I experienced it up through and including my PhD postdoc. The reason I have a cartoon of a chromosome here is another aspect of synthetic biology. It's often the scale at which we're attending. So here I'm referring to the sense of complexity. Some of you are aware that research groups have synthesized the entire instruction set for simple microbes at this point. The J. Craig Venter Institute accomplished that several years ago, and were able to show that they could transfer that whole synthetic piece into essentially the shell of a related organism and boot it up. Similar work is ongoing now in yeast, which is a significantly larger genome, but anticipated to be complete in some small number of years in the future. And so again, the scale and ambition of synthetic biologists is significant. One of the projects I'll tell you briefly about on my own has to do with attempting to rewrite the genetic code of a simple organism, a project that at the beginning I and my collaborators were told by several respected scientists, it's a terrible idea, you're not going to get it to work, it's too complicated, too many different things are going to go wrong. thankfully, they were mostly wrong, and we were mostly right. And there's important devils in the details there. And so when I think of barriers in synthetic biology, I just wanted to throw out some thoughts here. There's the technical barriers, and then there's societal barriers. And I'm going to touch on each of those. There's no way I'm going to cover the kind of depth that we all might like, but I am really interested in the conversations we might have. On the experimental side, one of the things synthetic biologists are trying to do is make it easier to get a lot done, to do a lot of experiments, because our design tools are still kind of poor. They're coming along, but they're still kind of poor. So one thing we need to be able to do is to try a lot of things, to do a lot of experiments in parallel. So if I have one element that I really need as a part of my design, the role of that element is to turn a gene on and to turn it on at exactly this level. Work with me on the abstract. Exactly this level. How much work does it take to be able to get that element, that on switch that works exactly here or kind of in this zone, but isn't way up here or way down? And so in terms of, and this connects to some of the work that I'll be talking about, the access, in fact, originally I had this as barriers to access, but I thought more broadly in terms of experiment, access to getting the tools in a larger number of hands, getting them affordable. There's the question of hardware tools, the ability to share designs and data across different sorts of efforts. I'm very involved in the iGEM competition, the International Genetically Engineered Machine Competition, which started here at MIT. And MIT is still quite involved in, and that's where undergraduate teams build across the world, about 280 different teams this year, build genetic devices, if you will. They're trying to program with pieces of DNA, some of which are available through the competition as part of a parts registry. So where the DNA can be shared, the DNA molecules can be shared. But even often more important is things like the performance data can be shared, the performance data that goes along with somebody's designs. So lowering the barriers for people to have access to good data from what's gone before is significant. And synthetic DNA, an area that I've been involved in, it's now, many of you are probably aware, but some might not, that it's now quite possible to simply design a piece of DNA from scratch of a known sequence with your C's, A's, G's, and T's, and just order it out. That's been possible for a very long time with short pieces of DNA, which we often call oligos, but increasingly the scales that you could just order from a company have gone from units of hundreds of bases long to tens and hundreds of bases long now to many thousands of bases long, and it's getting more and more routine. Now on the barriers to acceptance side, those of us who are doing the technical work often have concerns about the public's acceptance of synthetic biology work and fears that people will have. And if you will, there's sort of a fear of fear. There's a synthetic biologist's fear of what fears the public might have. And it's a worthy conversation in and of itself. How justified is that fear? But some of the things that we would hope that would get across, and it may be less of a challenge of this audience, I don't know. But part of it has to do with the challenges of logical thought processes. And a couple of slides, I actually have examples that are from not the synthetic biology. But, one of those big areas has to do with an understanding of risk. And this is, I think, a major societal challenge we have, not just a synthetic biology challenge. Which activities do we associate with what kind of risk and fear, and are we thinking reasonably about it? My wife and I, as parents, have this conversation all the time with respect to our children. Is it a bigger risk to them to let them skateboard, or is it a bigger risk for me to drive them around in my car? Within this whole regime, there's a lot of complexities behind the arguments of risk and fear. And I don't think there's easy solutions. I don't think the sound bites are going to do it. So one of the hopes is that everybody can appreciate that the complexities in this are worth trying to understand, and not just go off of somebody else's sound bites. In addition, the idea that biology in and of itself doesn't have to be alien or scary. An alien or scary factor influences our perception of risk in interesting ways. But I also want to touch on needs that the researchers have, and things that perhaps folks in my world, in the experimental world, but also do a better job of accepting as well, that might move forward. One of those is a lot of us don't like the idea that our work needs to be evaluated from the outside. It's easy for us to consider ourselves the ultimate expert as to the risks involved. And we are generally far better informed than most other people, even other researchers who aren't involved in our area, but that isn't an absolute. And so from my perspective, there is a need for a conversation between synthetic biologists and regulators and the public. It's also important to note that our work does receive different kinds of scrutiny. For those of us at universities, we have environmental health and safety evaluations that are actually quite in-depth and specific and are meant to serve that role. So one, the work sometimes needs scrutiny, and two, the work actually does receive a significant amount of scrutiny. That for certain applications, maybe not for everything, some regulation is likely to be necessary. I think in 2 weeks, when George Church and Ken Boyd talk, there's going to be an excellent example to think about that question. And also that the public needs a voice in these conversations. I've had colleagues that have done a great job of pointing out that it's easy to want to preach from the pulpit and say, here's the information we have to share with you so that you are more enlightened and less ignorant, that you can be more like us. And I think that's challenging. I don't think that's quite the right model. That, the conversation needs to go both ways, and those of us up here need a chance to ask questions and listen as a part of that conversation. Okay, with the time that I have, I'm going to zip through a few things. I'm actually going to go very briefly through the technical side. This is a slide relating to my own work. What this is a system that we call the BPU. It's in development. The biomolecular prototype is used. And the key elements here is that we are seeking to accelerate the way we can do experiments through a combination of DNA synthesis on the front end and putting that synthetic DNA to work in this component. It is a microfluidic platform. I'm not going to go into what that means, other than very small, very dense. This is on the surface of a microscope slide about the size of the phone. But the idea being that the user wants to be able to pose a question that can be phrased in the form of DNA. So that DNA needs to be synthesized, and then that DNA needs to be used, and you want to get that performance data out. This is a model of what basically everybody does in some form or another. What we're seeking to do is integrate this into a tight microfluidic process so that in many cases these questions can be asked in a day and answered in a day or two, at least the simpler ones, in a day or two, as opposed to weeks and months and longer. Now, I planned some particular technical examples, and again, in the interest of time, I'm going to quickly gloss over these effectively. These are 4 areas that we are applying that same pipeline to, the ability to ask questions in the form of DNA. One of them was Ron Weiss here on the MIT campus, genetic circuits that are designed to identify and hopefully someday treat cancer. In a different application, exploring an individual's complex spectrum of HIV mutations so that a better drug regimen can be determined. Designing enzymes that break down organophosphates, which specifically come as pesticides, and chemical weapons, a category of nerve agents. And the one example I did flesh out in here, an engineered genetic code, which could provide unique features to an engineered organism. So I'll tell you briefly what I mean by engineered code. And particularly the genetic code, and again, I think we've got a lot of biology background. I'd be happy to answer follow-up questions later. We know the DNA language can be simplified as C's, A's, G's, and T's. We also know there's a lot more complexity in it. But the language that's used to build proteins encoded in C's, A's, G's, and T's uses 3-letter words. So you can have 3-letter words A, G. There's a whole tape. 4 times 4 times 4 options gives you 64 of these 3-letter words with all those code elements. And at the end of the day, what these are used for is to represent the building blocks, to essentially instruct the machinery of the cell how to build this complex polypeptide, amino acid, individual amino acids. It folds in proteins, and proteins do most of the work inside our cells, and highly simplified. What's in between these is the table of those 64 words, 64 3-letter words, and how those words individually map to these building blocks. So you'll hear the term genetic code used in a lot of sloppy ways. In the strict technical sense, this is the genetic code. And importantly, it's nearly universal. So it's interesting that it's nearly universal in almost all living systems. It's also interesting where there are slight exceptions to that in nature. But think about this for a second. A virus that infects any organism is dependent on the genetic code of its host. The virus carries so little of its own instructions with it. It carries the instructions for making more of itself, but it doesn't carry the machinery for making more of itself. It's entirely or almost entirely dependent on the genetic code of its host. And so in the project that we've been working on with these great collaborators, Harvard Medical School, MIT, and Yale. The idea is that engineering a cell to have an alternate genetic code would mean, in principle, that the viruses that formerly infected this species could no longer infect it. It would also mean that in terms of sharing DNA-encoded functions with a wild, natural version of that exact same species, even though you could move pieces of DNA around from one to the other, if they were encoded using a different instruction language, each organism would not be able to fruitfully use the instructions from the other. So sometimes we refer to this concept as a genetic firewall, that the function could not pass through. So we have essentially accomplished this in a series of publications we've put out over the last couple of years. In the simplest possible way, we've taken one of those 64 words and removed it entirely from the genome of a simple organism, E. coli laboratory strain. And we've demonstrated some modification to its vulnerability to viruses that infect the bacteria. Now, the concept that we are considering, if it can be achieved, and it's much more complex to achieve this in higher organisms like plants, but the possibility, the potential value should be fairly obvious. If you could produce crop plants, if you can engineer those so that they were no longer vulnerable to viruses, that would be one major avenue of crop loss that you would have eliminated. And similarly, if you're concerned about the spread of genetically engineered functions, the different genetic code would impose this rule that pollen, for example, from this highly engineered species, if it were to make it out into the wild, would not be able to fruitfully pollinate a wild version of that same species. Okay. So that's some of the technical aspects that I wanted to touch on. And as I finish up, I have a few thoughts that I can go through quickly about risk. And one of those, really the theme throughout all of those really is learning how to think. And this is something that challenge not just you with, but also myself. Whenever I have a knee-jerk reaction to an argument that opposes mine, I have to challenge myself. So this is a great example of how people think about risk. I think we all can recognize in our culture. Oh, don't freak out about Ebola, but everyone does. And scientists say, oh, what you really should worry about is climate change, and from a very large fraction of people, not everybody, certainly not everybody. The response is like, oh, you gotta be kidding me. That's too abstract, it's not real to me. This is real to me, and I'm terrified of it, even though the chance of it affecting me is modest and quite enormous. And so one of the things that I like to advocate for, even though it's not my technical specialty, synthetic biology, is encouraging people to think about risk, and especially when it comes to early stage education, elementary school, junior high, and high school. I was not taught these things, and I wish I had been. I see my children being taught them a little bit better. Something simple along the lines of, hey, you're in history class, what do you think of the source? So I was never taught to question the source, and my children are being taught to think more about it. So here, which of these organisms do you think is a greater risk? Who thinks this one is the greater risk? Who thinks this one is the greater risk? All right, I see a few more hands and some other people who are waiting for me to finish up so they can listen to Jeff. Thinking about risk. Okay, so anyone know the name of this guy? I bet somebody knows the name. Vampiroteuthis infernalis, the vampire squid. It's hard to have a scarier name, and there are some scarier organisms, but this is not a sea cat, by the way. And this is the blue-ringed octopus found off the coast of Australia and some other places. Some friends of mine at the EPA pointed out to me that often risk, in a formal technical sense, like if you're trying to do a calculation, is thought of as probability times damage, the chance of something going wrong versus how bad that thing going wrong is. So it turns out that your chance of encountering the blue-ringed octopus is very low, but if it bites you, you can die. They have enough venom to kill about 30 people. There's about 4 species. So don't be terrified. Obviously, somebody thought it was a good idea to hold this in their hand. That's a different side story. But Vampiroteuthis, one, you're never going to encounter it unless you're diving, say, to 1,000 meters below the surface, because it lives in the dark and depths. And there's no known harm associated with it other than to its very small prey that it goes after. So all of this, this is all our layers of fear and association with color and everything else. I know I'm just about finished, but this is another example here. In terms of critical thinking, this is a hot topic item. I'm not going to tell you what I think of this issue, but in terms of evaluating a source, you look at this source here, which is in the Pinto car recalled after 27 deaths. I think there was a particular batch of Tylenol that was recalled after 7 deaths. You can still get Tylenol. Handguns, 32,000 deaths a year. Can somebody point out the logical problem with this argument? Not say whether you like the argument or what side. Anyone willing to point out a logical problem with the point this is trying to make?
First two are branded.
The first two are specific brands, and this one is referring to a whole gigantic category. So that's certainly one example of something that's category. What's that?
Category error.
Yeah, absolutely. No, this product has a very different function than these products.
If you said cars, generally, the number of yes and no.
That's right. In fact, that's what this graph is. I'm not saying I think particularly highly of this graph or poorly of it, but in terms of a source, I think most of us can appreciate, okay, this tells me where I can go and look for that information myself if I want it. There's more database. This is in fact comparing motor vehicle deaths and firearms. Of course, to really understand whether this is meaningful, it's worth finding out. Well, how do they tally these? Is there a source that that there's an article that goes with it? You get more of that information. And so my encouragement there is, and I'm not concerned about the thought processes of any one person here, but all of us, including myself, our thought processes are worth challenging again and again and again when it comes to risk and evaluating what a real risk is. And this is the one that I'll close on. You can't even read this, but there's a lot of different collections out there. Basically, what is your logical fallacy? What is a logical fallacy? So there's popular one somewhere. But the idea of the trouble that we get ourselves into comparing apples to oranges sometimes in our arguments. And so there's a lot of things that my kids are being taught in school that I think, to me personally, are less important than teaching them to think about these things. And so for those of you, particularly who are educators or thinking about that, I encourage you to think about how to fold that into your own aspect. And ad hominem attack is often simplified as the idea of an attack on somebody's character instead of an attack on an argument. And so I'll close with that one just because it abuses me. So, thank you very much. Sorry for going a little over time.
Thank you very much, Peter. That was a very entertaining talk. I really enjoyed it. So up next, we are going to have Jeffrey Way come and share some of his work and experiences. And then after Jeff's talk, we will be having small group discussions and I'll be leading people through that. But at this point, let's hear from Jeffrey.
Okay. Is my voice coming through? All right. Okay. Pete and I really are in the same field, and I was lucky that he went first because he gave my introduction, which I had not really prepared. So I'm going to talk about one example that illustrates a lot of what Pete was talking about in a general way. I'm going to talk about making a particular kind of class of therapeutics and diagnostics that have been going on with myself and also Pam Silver, who I work very closely with at Harvard Medical School, who is also my wife. And here's what we do. We have right now a particular grant from DARPA that is in their BRICS program. This stands for Biological Robustness in Complex Settings. And basically what they want to do is have people engineer organisms that can really function in the wild. A lot of synthetic biology up to now has focused on engineering lab strains of E. coli or lab strains of other organisms that function in a lab environment that's very controlled. And what we did recently, which I'll tell you about, is to actually take the wild bacteria that we just isolated out of a mouse, engineered it without really knowing a whole lot about it, put it back in the mouse and get it to behave. But there are a bunch of challenges associated with some which we alluded to. The opportunities that we can engineer bacteria to perform useful tasks. And the one thing that we have in mind is by a bacteria that could diagnose gut inflammation. So the idea here, the vision here is that, let's say you have a gut disorder, you have inflammatory bowel disease, and your gut is going through states of inflammation, and sometimes it's bad, sometimes it's okay. It depends on what you eat, depends on how you're behaving. And what you do in the synthetic biology world is you take some bacteria that lodge in your intestine, and then depending on how inflamed you are, it will sense the level of inflammation, and then when you poop out those bacteria, you will get a signal. The toilet bowl will turn green or something like that, easy to read, and you can then figure out, should I go change my diet, should I go to the doctor, what should I do? But this brings up, again, some of the problems that Pete pointed out of what about environmental contamination? Now you've got a genetically engineered bacteria sitting inside someone's intestine, it's getting pooped out into the environment. How do you contain it? How do you prevent its genes from spreading further, and so on? And so we want things to function in diverse environments, such as the intestines of a whole bunch of different people, but then we would want them to die on cue. For example, once they get pooped out, they should all die. Now, Pete also alluded to biological calculators. The point that and this is something where we were successful, and actually it helped us get a big bolus of money from DARPA. So I'll just tell you this one scientific story. Even though we are very, very far away from doing what computers do, the thing about live organisms is that they can live off the land. And so you can engineer a very simple genetic circuit, but then you can put it in all sorts of diverse environments. And most of what you want about the organism is the fact that it lives in your gut or in a desert or in a Great Lake or wherever, and it can just live there. And then if you are clever about the genetic engineering that you do, you can get it to sense something in the environment and tell you about it and perform a very simple logical calculation. And the point is not that it's doing something complicated, but it's doing something almost for free. It's, like I say, it's just living off the land, absorbing sunlight or whatever nutrients are out there, and then performing some function on your behalf. And so an example of the kind of simple genetic circuit that we deal with is here. This was something that was conceptualized about 15 years ago in the lab of Jim Collins, who's now at MIT, who was at BU at the time. This is the so-called genetic toggle switch, where you have a piece of DNA, a repressor that is made from part of the DNA, and it turns off the other gene for another repressor, and then this repressor turns off this one, and so it can exist in a bistable state. And then you have a recorder here that is made along with one of those repressors, so you can tell what state you're in. And now, by way of reminding you of what we're dealing with, here is a piece of DNA that corresponds to really just about this much of our genetic construct. And as Pete also mentioned, we can now just buy these pieces. This whole thing would fit on a piece of DNA, probably about 2,000 bases, and that would cost, right now, depending on where you buy the DNA, 15 to 30 cents per base pair. So 30 cents per base pair, that's about $600. So that's not bad. If it saves you a month of a graduate student's time, it's well worth it. It's an easy economic trade-off. So you can just buy that piece of DNA, but you do have to be able to design exactly what you want to make. And so what you're really looking at is a piece of DNA where you cobble things from nature, and that's what you're literally going to order. And that's this is probably, well, we can count how many turns of a base pair. It's 10 base pairs per turn in a piece of DNA, so that's about 100 base pairs. And then you have to say, well, I want a protein to bind to a certain part of this DNA, and this would be one of the repressors that turns off the other gene. And then the way all that is going to work, and that would be part of my construct. And then part of what that works, how that works is it interacts with the host proteins. And in this case, it's got to work with RNA polymerase, which is the thing that actually makes an RNA copy of the gene, makes the messenger RNA, and then that gets translated into a protein. And then you're kind of off to the races. Then that protein would be your repressor that would define one of the states that you're in. And so when we actually do this on a day-to-day basis, at least I spend a lot of my time looking at structures like this, thinking about where I'm going to put things on DNA, where are different proteins going to bind, and so on. But for most people's purposes, you can just abstract this as that, something that looks kind of like an electrical circuit diagram. So that the toggle switch was developed by Jim Collins' group. The trigger memory system was developed in Pam Silver's lab a few years later. And the idea here is that you have an activator that responds to an environmental signal. So you've got one of these promoter elements, one of these things that responds to the environment. And then you make it, by genetic engineering, you associate that with a protein that can then change the state of a memory element. Okay, and you can arbitrarily associate these things. And then recently we put this all together into an integrated system where we have a toggle switch, which is extremely stable. That's the wonder of this toggle switch, is that it doesn't change state by accident. You really have to push it. But if you have a switch element, you can start off with your system in one state, and so the blue guy here is on, it's turning off the yellow guy, and the yellow guy is not there, so it's not turning off the blue guy, and so you have that stable state. But then when you come in with your environmental signal, then the switch element will come here, turn off the blue guy by default, then the yellow guy goes on, and then it keeps the blue guy off, and so now you permanently switch states. And we actually put this into practice and published this last year sometime, where we basically stole from nature, and this is really the secret of synthetic biology, is that you don't really make this stuff up from scratch. You find things out of nature that do roughly what you want, and then you steal them, and then you splice them together. And that way you don't really have to understand in excruciating detail how every atom of your system works, because people just aren't that smart. So what we did was to steal from a bacterial virus a naturally occurring toggle switch, which is defined by these proteins C1 and CRO that are from, there's a whole biological story about how these were discovered, but you can take this chunk from a virus associated with a drug resistance, put it in upstream of a gene, LAC-Z. This goes for beta-galactosidase, which breaks down lactose. We can have a colored dye that is a substrate for the enzyme made by this. And then we have a trigger element where anhydrotetracycline, a cheap tetracycline derivative, will be used as the environmental stimulus to turn on this CRO protein. And so we can switch from the C1 state to the CRO state. When the CRO state is on, you make this enzyme, and then basically the E. coli that you're sitting in will turn indicator plates either blue if they have a blue dye that responds, or change the color of a colony on a kind of a classic medical indicator plate. And I should say these McConkie lactose plates, these have been in use for 100 years in medical labs. So this is very, at the biological level, this is very low tech. We do some fancy genetic engineering, but really at the end of the day, you look at bacterial colonies on plates using 1930s level technology. As far as everything except the actual manipulation of the DNA goes, but we use microscopes, plates, and so forth. So with this memory system, we did have to test that it actually works. And the way you do that is to take a tube of your bacteria, and you either treat it with anhydrotetracycline or you don't. And for a couple hours, for a good 6 hours, bacteria grow in the presence of this trigger, this environmental signal. And if if it's present, then they switch state, and so they make blue colonies. And then if you take these guys, plate them out, they have not switched state, they make white colonies on the indicator plate. The blue guys, then, you either grow them continually with the signal, and so they have to stay in that state, or you take away the signal, and then you grow them for up to 5 days. And basically, when they switch in the first place, within about 4 hours, which corresponds to a couple cell divisions, 4 cell divisions, they've all switched. But then you take away the signal, and here, okay, you grow them for 5 days. That's a 1,000-fold dilution every day, corresponds to about 50 cell divisions. They more than 90% remain in the switched state. So for environmental testing purposes, diagnosis purposes, this is fine. That's plenty good enough. So what did we do? What we did was take those bacteria and put them in a mouse. And the experiment was fairly simple. You have your mouse, you give it the bacteria, and you have to kind of do this to the mice, and they don't like it. And I got bitten a couple of times by not holding the mouse carefully enough. But the bacteria will lodge in their gut, and then a day later, you let the mouse recover from the whole procedure, and then then what you do is you give them anhydrotetracycline and the bacteria in their guts as determined by what they poop out. So then we just, this is really glamorous work, as you can tell. We go into the mouse facility, and I will say that the smell you get used to really quickly. But you go in and you collect their fecal matter, and the good thing about mice is that they poop almost on cue. If you grab them out of a cage and put them in a little box, they will poop within 2-3 minutes. Take them out, collect the poo, shake it up to separate all the bacteria from the fibrous matter, and then you plate it out on these same indicator plates, and really, the results were unambiguous. 100% of the bacteria switched. And now you can say, well, who cares? That's nice that the whole thing worked in a mouse. It's a more rough environment. But basically, you're using the mice as a giant incubator. Basically, you're using the mouse as a giant test tube. And I don't know, are you saying that? Or are you saying this is cool? Maybe it was obvious that it was going to work, or maybe it's cool. One thing that has happened, and this is one of our colleagues at the Wyss Institute, has recently done an experiment where she was studying, and this is really recent and not published, so don't tell the whole world. But basically, if you put this in a wound next to a metal implant, these same bacteria, they will set up a festering wound. And the basic issue is that the underlying scientific issue is that when you have something like a knee replacement or a hip replacement, you have a piece of metal or plastic in your body, and the blood supply around those things is not very good. The blood supply is obviously not going into the metal. And so bacteria get on the metal surface. It's not clear that blood will deliver either antibiotics or immune cells or anything else to those bacteria, and you often will set up these persistent infections around implants. And so with something like these bacteria, what we should be able to do is to implant those in the wound and then give the mouse tetracycline and ask, can the antibiotic even reach those bacteria? Are they in a protected environment? And the best way to tell is with something like this memory device, because then you get the bacteria out and you shake them up, and then you put them onto an agar plate that is one of these indicator plates. They grow into colonies, and you can tell what they experienced at some time in the past. So this is an example of a very simple bacterial calculator that can function out in the real world and tell you something that otherwise is very hard to figure out. So, just to sum up what next, one of the things that we're doing right now and starting to have success with is to use, instead of using a chemical signal where we just dose the mouse, and again, you could say the tetracycline thing, well, that was, of course, that was going to work because the bacteria are in the gut, and then you're putting tetracycline into the drinking water. So, of course, the tetracycline is going to be there. But remember about that trigger element, you can have a genetic control element that responds to basically anything that you want, anything that nature can respond to. You can take the control elements, put them upstream of your gene, put them into your circuit, and then you can get your circuit to respond to that thing. And so we have bacteria now that respond to inflammation. When you have inflammation in your gut, there are certain characteristic chemicals like reactive oxygen species that get made by the host cells, actually as a way of killing the bacteria. And then the bacteria have a whole defensive response against things like hydrogen peroxide, basically. And when that gets turned on, genes get activated. We can hook our system into that and say, all right, this bacteria that just got moved out, it was experiencing inflammation when it was back in the gut. And like I said, we can then use that to decide, is this person, does somebody with this problem have inflammatory bowel disease? Maybe, and we've actually done experiments where we've engineered one kind of bacteria to have this sensor and memory system, and then we would put in a normal bacteria that is just salmonella, basically. It's something that will cause gut inflammation in mice and ultimately kill them. It causes typhoid fever. And as an early warning, the engineered bacteria will say there is serious inflammation in the intestine of this animal. And so that can be used for early warning purposes, or if you were really clever, and this is where we have not gone yet, you could engineer the bacteria to do something about the guys that are causing inflammation. As a final note, we are also, and then there's all sorts of other clever things you can do. We are trying right now to count cell divisions and count time. One of the things we've done and published recently is, in terms of counting time, is that photosynthetic bacteria have a natural cycle of 24 hours, and they actually have a set of 3 proteins that will oscillate in the state, in their phosphorylation state, with a 24-hour period. And you can actually just take the proteins and put them in a tube with ATP, the energy molecule of the cell, and they will oscillate with a 24-hour period without anything else. You just keep adding ATP and they keep oscillating. And we have put those from the photosynthetic bacteria. It makes sense why they have that, because they're totally dependent on sunlight, and so you want to make one set of things when it's light out, another set of things when it's dark. We transplanted that into E. coli, and now we can get the E. coli to cycle, and so there's the beginning of a timing system there, and then you can imagine hooking that up to chemical sensors and say, well, how long ago was the inflammation? How long ago did the bacteria see tetracycline? The final thing that we are doing is trying to recode some of these organisms, like Pete was talking about, change the genetic code. And there we are just starting, but we are trying something more aggressive in terms of the number of changes we're trying to make in the bacterial genome. So this is where, like I said, it's affordable to make a couple thousand bases. To really redo a bacterial genome, what we are planning to do is make about 30,000 changes in a 4.5 million base pair unit. And so we have to actually buy synthetic DNA for that 4.5 million bases, and then it becomes an economic problem. You've got maybe, if you're lucky, you could maybe get 10 cents a base, and then it's only $500,000, only $500,000 to buy the genome of a new organism. So I will let everybody ponder that and open it up for questions. Questions? Yes.
Could you make a a switch to kill bacteria in case it's like you wanted to then get it out of that system?
Yes. Yeah. What we are thinking, and actually, I don't know, Pete, you want to come up? I don't know. You know, is this how it's supposed to go?
So we're actually going to hold off on questions for about 15 minutes and have small group discussions at the table. So jumping down a little.
Okay.
But
Can I answer his one question? Okay, because I could have mentioned it actually. For example, in the specific case of bacteria that are going to go into the gut, what we plan to do is engineer them to be oxygen sensitive. In the large intestine, it's really an anaerobic environment. And when things get pooped out, there are two dramatic changes. One is that the temperature drops from body temperature to room temperature, and one is that there's all of a sudden oxygen. And so we think if we can combine those two signals, then we could really get them to die on cue. And that's going to be important in terms of, again, making the public comfortable, thinking about, you know, on the one hand, the inflammatory bowel syndrome is a terrible disease. It's really bad. But how do you weigh the benefit to a set of individuals with the risk of environmental contamination that could affect everybody maybe in a mild way, maybe in a serious way? Okay.
Wonderful.
Go amongst yourselves.
Exactly. So our next stage is to have small group discussion at the table. So we have some volunteers going around handing out iPads. So the iPads are set to Twitter. So if you have questions, if you tweet the hashtag MIT Soapbox, it's all the triangular pieces that are on the table, in case you don't remember. They'll be posted on the screen, and then we'll be having a large group discussion. You can ask questions to our visiting scientists. You can ask questions to each other, but we want to start a conversation. And we'll take about 15 minutes for that.
What? Do I join us? What? Yes.
Yes.
Chemical part.
Literally and yeah. But
They have like basic protein expression of so that's so take the light out. Still out. So one of those kind of like space bikes. Because it's not that strong. So it's not it's not a thing. It's like it's not fun. It's not like fire stuff. So you can run a legit school. So it's I'm not really affiliated with owners. They just kind of let us walk like math science set up. Yeah, so we just use that. Sometimes you're in and out of just yeah. It's a personal experience. Something will be pulled off, like, given my actual this kind of stuff. So, I just want to help people get out there. So, if they're interested, yeah, I mean, sometimes they're also very creative. Yeah, sometimes, so like down to single cells in between. The whole dish actually really is great. It's taking advantage.
So we'll be gathering this within about 2 to 3 minutes. So start wrapping up conversations, tweet in your last questions. But we'll be starting in just a few minutes.
So,
So I can think about these questions. Oh, that's okay. I think now, well, now we're just break down all these questions.
All right, so it seems like I've managed to kill the conversation. So let's come back to a large group discussion. So just so you guys maybe can keep tweeting in questions, we're going to be taking questions from the audience. So you guys can raise your hand, we'll come around with a microphone and you can ask. Or if you prefer to be anonymous, you can tweet your questions in and we'll go through these questions as well. We have also been getting some questions from our internet audience also. So it seems to be getting to be a good conversation. One quick thing, we'll also be passing out some surveys and we really appreciate it if you would fill them out. Give us some feedback about tonight. If you do add your name to the survey, we are raffling off an MIT hacked mug. So a lot of tonight was about breaking barriers and overcoming challenges, just like putting a car on the top of the MIT Dome is overcoming a challenge of gravity. So we will draw one winner from tonight for filling out your surveys also. But with that, let's get to the question sessions. Why don't we actually just start by taking one off of here. So let's actually start this first one. So what is the risk in letting pooping bacteria run away? Say that.
All right.
squarely in your wheelhouse, all right.
Okay. It depends on what the bacteria are, and that's not really an easy question to answer because it really depends on what you've engineered into the bacteria. Presumably, the bacteria themselves are pretty innocuous. Otherwise, you're not going to be putting them into people. And then the question, if they're GMOs, these bacteria are genetically modified organisms. And the question is, how do you feel about them being out in the environment, exchanging DNA with other organisms? The DNA that is engineered that we're putting in them is really never going to confer a selective advantage. And that's part of the thinking, the process that you have to go through when you do this engineering is, if this escapes, will it make a bacteria more pathogenic? Will it become an ecological problem because it confers a growth advantage to the bacteria? And the answer really has to be no, or else you never release it. But then you still might worry, well, you know, I've got this artificial construct and I put it into these guys and they're getting released. What could happen? And it's just impossible to answer, to be honest. The answer is probably nothing. And also, there was actually exactly this debate at the dawn of the recombinant DNA era, and it's hosted on an MIT website someplace. There's a documentary about some of the Cambridge City Council hearings called summarized in Mark Matashley's phrase, hypothetical risk. And there, the mayor, Al Vellucci, who was mayor of Cambridge, was saying, you know, can you really prove that this stuff is not dangerous? And the answer is you cannot prove a negative. But realistically, we've had recombinant DNA for a good 30, 40 years. Nothing bad has happened to the environment unless you want to consider GMO crops bad, but that's really a commercially driven thing. But nothing really bad has happened because of environmental contamination. And there certainly has been environmental contamination because in the early days, people actually used to throw a lot of stuff down the drain. There's another question up there, how do you dispose of this stuff? What we do now is we put it in special bins that then get autoclaved. And so everything is killed before it's put out in the environment. But autoclave means it's kind of like a giant pressure cooker. It's a giant steam-driven thing where you heat something up to higher than boiling temperature under pressure, and everything gets sterilized basically. So everything that leaves the lab gets sterilized. Now, when I was a graduate student, we didn't really do that. And we would dump stuff in the trash or sometimes drop it on the floor or spill things on the floor. That happens. Stuff gets spilled on the floor, you walk around, you're not aware of it, you drag it out of the lab, that kind of stuff happens. If you are dealing with anything that is dangerous, then you don't do it in a regular lab. You have booties on and you actually go through all sorts of containment procedures. But the really dangerous things that everybody worries about are natural pathogens. That's what you do with salmonella, that's a human pathogen, or Ebola virus, or things like that. Those are the really dangerous things. And what we do with recombinant DNA really pales in comparison.
Any questions from the audience, or should we keep going from Twitter?
I have a question. It's kind of a vague question. So the question's kind of vague, but how long is it going to be before we have, like, instead of someone having a natural microbiome, digitally engineered microbiome, where it's like, you know, doctor bacteria like controlling it and upregulating and upregulating other cells, and they're all synthetic.
Is that going to happen? I would say that should not happen because there's too much that we don't know, and actually nature works pretty well. The question is, but person, if you have inflammatory bowel syndrome, then maybe you want a doctored microbiome. And then for people like that, then you probably do want exactly what you're talking about.
I just wanted to add to that. I think if you look at the distinction between a completely engineered microbiome versus a microbiome that has an engineered component in it. So as soon as you engineer it and it's running in your own gut, you're still being exposed through all of your various pathways to the natural components that are going to be involved and compete. So it's unlikely to stay engineered in a pure sense for very long. But the potential for I'm suspecting over the next couple of years we're going to be seeing applications for trials.
There will be clinical trials in the next couple of years.
So we collaborate. We have a collaboration starting here at Long where we're looking at composed microbiomes. I think you guys have some work in that area as well. And so there's a version of it where you do a certain form of genetic engineering where you're engineering the level of picking your community members. You might be taking natural species and not engineering them genetically, but you're engineering the community by your choice of composition. And then there's the additional layer on top of that, which is, of course, altering the genes inside. So I think we see applications in both areas. The first of those, the engineered composed communities of natural strains, is going to get through regulatory approvals much more quickly. So you can see this interesting progression, I think.
Right.
It's going to play out especially over this coming decade.
Let's do one from here, and then we'll get to your question. So would it be possible to change the meaning of every codon in an organism?
So this is something that I think a lot about. It is theoretically possible, but the difficulty of that is extremely hard to evaluate even with kind of theoretical estimates that we do. There's a phenomenal amount of engineering that you would have to do, including the intermediate stages of getting to that final completely shuffled state. So it's theoretically possible. We understand enough of the mechanisms involved in which enzymes are involved, the molecules in play, but so you need to not just change the DNA. This is one of the important parts. You need to not just change the usage of your new dictionary, not just change the DNA, but you also have to change with it the machinery, which is a combination of protein and RNA-based machinery that reads and implements the code. And so that also would have to be re-engineered and rewired corresponding.
Yeah.
So it's an exciting idea, but it's a real.
It would be difficult, and it underscores. Actually, I'm going to steal from I think one publication that Pete was a co-author on from George Church's lab, where they actually tried to recode a set of the most important genes in E. coli, a set of genes that make the ribosome, changed all the codons, made what are supposed to be synonymous changes. So you have a codon there are 64 different possibilities that Pete mentioned, but there are only 20 amino acids. So there's a lot of redundancy in the genetic code. And so you could, if you want to put in an amino acid like glycine in some position, you have 4 different ways of doing that. And you could substitute one codon, one set of 3, for another set. And what Pete and George Church and people did was to make a bunch of synonymous changes, and the organisms really did not grow very well, even though the protein sequences were not being changed. And that's because when you make the messenger RNA that codes for the protein, it has certain ways that it likes to fold up. And so you're also changing that. And we have an impression from teaching classes and stuff that biology is modular, and it kind of is, and it's easier to understand because that's how the human brain works. And everybody says, oh, it's just like computers, biology is just like computers, and we have these genes and they come in nice little blocks. But part of that is an imposition of the human brain, and that's how we understand things to work. That's why computers work the way they do. They've been adapted to our brains. Biology really hasn't. And so when you go in and change some of these things, you're changing more things than you imagine, and then you find out, you test it and it doesn't work as well as you hoped. So I think it is actually, it's going to be a big challenge, basically.
Question here, right?
Thank you. It's a question to both speakers, and it's about participation. And the 2 things I imagine is by participating either with scientists like yourself at the level at which you've heard the work that's unfolding in your research, or participating at other levels. And I'm an environmental and resource economist. I work with the World Wildlife Federation. I have a personal interest going forward to begin to work with people in the field, in your field. And so it's a question asking, and I'll give you an application, for example. This morning at Boston University at the Barney School, I did a talk about agricultural microeconomic models, and asked what's going to happen to cotton and wheat and other crops going forward in the next 25 years. Now, if one began working with scientists like yourself, would an environmental and resource economist work with someone directly with you? Would that be able to generate certain kinds of chemistry that are and results that are useful? Or would they better be working with fertilizer or pharmaceutical or other companies that are modifying crops and looking at food security issues with them probably?
I think my initial answer there is the potential to work with the academics, pseudo-academics, calling myself a pseudo-academic,
Right,
that's
yes.
interesting space. I think that's going to be increasing over time. So if you looked 5 years back, I'd say there were less potential for that kind of interaction. Still potential for interaction, having very fruitful conversations and kind of informing each other, but perhaps less so in terms of like undertaking a research project. But increasingly now, as synthetic biologists are getting closer to those applications that would function out in a more open environment, or have to think about, like Jeff was talking about, what impacts they might have in the environment. I think there's more and more of a need for those interactions. For the people in the different specialties to learn from each other. Some colleagues of mine, especially Ken Hoy at MIT here, who's done a great job of bringing people together to have some of those initial conversations that I really value being a part of and I've learned a lot.
Depends very much on the stage of the application.
Yeah.
Where it's reached.
Right. I will say, I mean, it's easy for people like us to talk about gut bacteria and medical applications, but food security is going to be a bigger and bigger issue for the world, because the way we've dealt with it since the 1950s has been through the so-called Green Revolution, which has involved basically a combination of traditional crop breeding, where you just cross things and do manipulate genetics and get things that are better, but not really genetic engineering, but also increased use of fertilizer and water. And so the Green Revolution was great. It increased per acre crop production in a lot of situations up to a factor of 5 or 6. But the benefits from that are kind of peaking out. Basically, the Green Revolution technology is kind of maxing out, and the world's population just continues to grow. And so there's a real potential problem. And also the nature of the Green Revolution is the more you use it, the less sustainable it is, because it really does rely on heavy fertilizer and water use. And so there's some thinking that genetic engineering is actually going to provide for the next Green Revolution, the next way people are really going to get food. The most partisan people on this issue are Monsanto, obviously, and you can think of them what you want, but one woman from Monsanto claimed to me that if you just calculate, so one thing that people need in their diet is omega-3 fatty acids, and those come from fish, okay? We don't make them, but our immune system is hardwired to require some level of omega-3 fatty acids to balance our pro and anti-inflammatory state. If we don't get enough, then we enter a pro-inflammatory state, just because icosanoids are made from. There's a whole bunch of biochemistry that I won't bore you with. But the point is that we need omega-3 fatty acids, and if you were to give everybody in the world a proper dose, you would fish all the fish out of the sea. So you're not going to be able to do that, and what you really want are genetically engineered plants that will make omega-3 fatty acids, so you don't have to fish all the fish out of the sea. And there are going to be things like that, just, where are you going to get the food for more and more people, and how are you going to do it without using ridiculous amounts of fertilizer?
At the granular level of your work, would it make sense for an environmental economist to stand by and see what you're doing? Does it make sense for them to intervene at a much more aggregated later
stage? I have no idea. Honestly, I don't know. I think probably where we really get the most benefit from people outside our field is telling us what to do. A lot of times, and pointing out problems that need to be solved. A lot of times, we are stuck in our labs and we know about cancer and immune disorders because those have been scientifically trendy to work on. But there are other things in areas of sustainability or just organ systems that are not so trendy, the kidney, gut, skin disorders. People who are synthetic biologists just don't know enough basic biology to know what to do. And things with plants and the looming human crises of global warming, what can we really do? We need someone to tell us what the problems are going to be.
Thank you so much.
So there's a question on this side here.
Hello. I was just wondering, has there been much work into whether developing genetically modified bacteria that respond to other genetically modified bacteria, kind of like networking them in a sense, or is that not a very explored area?
I think it's been explored a little bit at sort of lab toy level. I don't mean toy in a derogatory sense, but a sort of exploration of what's possible. So
Right.
There are ways that bacteria can pass DNA back and forth between each other under some circumstances. One of those mechanisms is called conjugation. It's literally a physical conduit from one to another. So I can't think of a specific paper off the top of my head, but I believe in a sense people have explored it, but I'm not aware of conclusive.
It's not yet still. Okay.
Great, let's take one more question off the Twitter feed. So how can we get the public involved and willing to engage in these types of ideas and conversations? We have an audience tonight of 54, but obviously there are more than 50 people who live in Cambridge, for example. So do you guys have any ideas or suggestions to continue conversations like these?
I would love to reflect that one back on this crowd here, which I think is probably about as adept at answering that question as I am. It's not such a technical question that it requires my expertise. I'd be interested in ideas that people have.
So, and then the second half of the question is, can the arts be used towards this end? And this has been one of the interesting evolutions within synthetic biology, is that, for example, there's this iGEM competition and everybody tries to make a clever genetic machine, and sometimes people are trying to cure diseases or increase food security or all these things that we've been talking about, but sometimes people just want to make art, and they will take genetically engineered bacteria and have them fluorescing in different patterns and have them talking to each other in unpredictable ways to make exotic patterns. And genetic engineering as a substrate for art is something that artists are now seriously exploring, and it's just fun. It's just really cool, and I think that is a perfect example of how to get the public more involved, because there are visual things that you can do. There are also things related to smells and chemicals. You can engineer bacteria to produce all sorts of chemicals, most of which smell. And so there's just a huge opportunity in the area of the arts. And also your engineering biology and biological systems kind of run the gamut from being beautiful to ugly, and with everything in between. And so there's just a huge substrate to work with for artists.
So I think we have time for 2 more questions.
Well, I was hoping that someone literally in here would give an idea of answering that question, if anybody was willing to take a stab at it.
Anybody have any suggestions on how to get more people involved?
It would be great if the public could do it as some genetic engineering, go out and see what it is, and how it looks like. I think there's a lot of magic around.
So a little bit of self-promotion. We have a group coming in here on the 24th into our Idea Hub space, and they're actually making an at-home, like countertop synthetic biology kit called Amino, and they're going to be letting visitors do some basic synthetic biology up on the 2nd floor of the museum on the 24th. A little bit of self-promotion, but I think that's a really great way to get people involved. Are there any other ideas from the audience? No? Yeah, so I think we have time for 2 more questions. Anything from the audience?
So my question was, are you concerned about potential abuse of intellectual property?
Sorry, I'm still having a little
Are you concerned about abuse of intellectual property? Monsanto, a lot of the reason why a lot of people have problems with Monsanto is not because of the underlying technology, but because of how people perceive it. These farmers, I certainly would be concerned if it could grow a plant because it was owned as I gave it feed and then it died, and that's why, you know, I can just imagine, I just worry that anything and everything becomes patented, copyrighted, trademarked, and then who knows what the limits of that kind of things?
Yeah, I think our society still has a lot of wrestling to do with those issues. When it comes to Monsanto, it's an easy one to hold up there. There are ways in which I'm convinced Monsanto is looked at a little bit like the devil's twig that I held up there. There are multiple sides to the Monsanto story, and it's worth looking at both of them. I've seen arguments and details of individual story on both sides that have given me a lot of pause for thought. Not that I'm not concerned about entities like Monsanto, of course there's others. I am concerned about the patenting of life and those rights being taken to an illogical extreme. I haven't seen what I think of that yet in terms of the Supreme Court rulings. I thought the myriad Supreme Court ruling was interesting. There was a lot of vagueness to it. That one had to do with when is a gene patentable, and they laid it down in some ways that a lot of us technically weren't quite on board with. It's like making a distinction, yes, no. Maybe that's in the eyes of the law, but it doesn't make as much sense in the eyes of the scientists. So I do share some of those concerns.
I actually don't. I was a director of intellectual property at the drug company where I used to work for about 10 years. And what I learned from learning about the patent system and passing the patent bar is, first of all, 99% of all patents actually never lead anywhere. It's actually a huge waste of time for a lot of people who file patents. And there are certain limits in patent law that prevent you from patenting things that exist in nature. If you have a new idea, then you can patent that, and it really has to be new product of your imagination and so forth. The real issue with patents that disturbs people, I think, is basically the bargain that the government makes with you. The bargain is you have an idea, the government gives you a monopoly on that idea in exchange for making the idea public. And in countries where that system did not operate, so for example, Imperial China, everything was a trade secret. If you look at the Japanese fermentation and soy sauce industry, everything was a trade secret. And so this information was not disseminated. And there's just a trade-off between disseminating information and then getting a monopoly on the information for a finite period of time. And that's a public policy issue. It's like welfare, you know. If you give people too much, they get lazy. If you give them nothing, they starve to death. Something in the middle is right. It's something that has to be constantly tweaked. But the fundamental goal of the system really is the betterment of the economy and people's welfare. And so I think that it's not something where there's an easy answer. It's always easy to point at inventions and inventors who have a lot of power because of the monopoly that they get out of their invention. But then you could say, well, they should share that with the public. And then if you look at countries where they don't have a patent system, they also don't invent stuff because the incentive is not there. So that's really a complicated issue.
So let's do one final question for the Twitter feed, and then perhaps if you guys still have questions, you might be able to ask them afterward. So the last question is, so what's the worst that can happen, and why should or shouldn't I be worried about?
Digesting that. Yes. I think the worst that could happen is that people, I think the risk of an accident is really infinitesimal. Okay, that's something that I really do not worry about. And by point of comparison, one of the things, I recently read a book called Command and Control, which was about nuclear weapons and just our nuclear missiles that were all aimed at Russia. And basically you had a nuclear warhead sitting on top of enough explosive material that it could be fired all the way to Russia, a couple thousand miles. And nuclear accidents really happen at a low level all the time. There would be a B-52 that would catch on fire that was carrying a weapon. that stuff was really scary. And there were a couple incidents that killed people in the United States, and there was apparently an incident in Russia that killed over 100 people. There was just a test that went bad. And the people were killed not from the nuclear aspect, but just from the rocket propellants that were sitting under the nuclear warheads. Fortunately, the nuclear warheads never went off, but the rocket propellants were a whole problem unto themselves. We are not going to have an accident problem like that. The most scary thing is that somebody engineers a clever death virus or death bacterium, and that is a genuinely scary possibility. The thing that makes me less worried about it is that, but it's still worth worrying about, is that even the simple stuff that I was talking about, like making a memory switch, that's just hard to do. And making something, we're not very good at biological engineering, and if you wanted to use biology to kill people, what you would really do is use anthrax or Ebola or something that is naturally occurring. But if you were devious enough, then you could use it for evil. And there, unfortunately, the genie is out of the bottle. I think the basic technology that enables you to do that is out of the bottle. What is really limiting is human creativity. We have a lot of trouble making things that are good, and it would probably be even more difficult to make things that are evil. But for all these things, we're limited by our own creativity and intellectual abilities.
So I think the worst that can happen, the long run, Jeff was also talking about, is that someone will use living creatures, microbes, pathogens to hurt other people. And we know this with almost near certainty that will happen at some point in the future. And the fact that the engineering tools are getting more accessible makes it more likely that there will be some level of engineering involved in that. And to look to the near certainty of that, all you have to do is look to the past, really, because people have done it in the past, first with naturally occurring microbes, and then with a certain amount of artistry for what was available at the time in the 70s, 80s, and 90s, with simple genetic manipulations. If anyone's interested, you can read up on the history of the Soviet bioweapons program to look at what they did with the technology available at that time.
There's a book called Germs that if you want to read about it, it's a very scary book. I don't know if you've read it, but Judith Miller, who used to be the New York Times, is the first author, and it's mostly about the Soviet germ warfare program, and they were really serious about developing death viruses and so forth. And there was, in fact, an anthrax outbreak in the Soviet Union that was just a laboratory screw-up, and somehow some of it got released, and it's been documented that the people who died had multiple independent strains of anthrax in their lungs, and so it was clearly for weaponized material, and it all came from a certain building. They were in downtown Sverdlovsk, where they were able to calculate from the wind patterns on those days exactly where it came from, animals that died and humans that died, and it was clearly from weaponization. Those were natural bacteria, but also part of their program involved evolving viruses. And again, like Pete said, that was the technology at the time. There was an example of one guy who died, and they harvested, because of a laboratory accident, and they harvested the virus out of it, and indeed it was more pathogenic than the rest of the viruses, because it had just evolved to become more pathogenic while it was in the system, so they isolated it and kept it going, and they actually weaponized that. And if you want to be an informed citizen, you should look into those kinds of programs, because that is really scary.
Well, thank you very much. Let's
On that note.
give a hand for our presenters. And thank you all for coming out tonight. Just want to advertise our upcoming ones, and perhaps over the next few, you might find out a little bit more about ways we could try to limit some of those challenges that are coming up, and perhaps make some of those risks a little less scary. So I hope you guys will consider joining us for the next 2 weeks. On the 14th, we're talking about ways to customize nature. in the ways we are able to engineer organisms. And then on the 21st, we're going to have more of a debate about policy and how and what types of policy should be enacted for synthetic biology. So I hope you guys will all join us again. We'll be doing the drawing in a couple short minutes, but until then, feel free to enjoy brownies and cookies and more tea. And then we will have a short little drawing for a hacked MIT mug to look at how you overcome challenges. Thank you for coming.